All right, welcome back, everyone. Let's just jump right into today's explainer. We've got a really fascinating piece of AI news intersecting with computational biology. We're talking about the brand new RegNetAgents multi-agent framework. We'll be exploring how it uses cutting-edge AI orchestration to tackle one of the most stubborn bottlenecks in cancer research, which is figuring out how to validate the actual genetic drivers of tumors.
So here's the fundamental problem. How on earth do we isolate true cancer drivers from just a massive sea of genomic noise? Honestly, finding actual driver regulators in these huge data sets, it's literally like finding a needle in a haystack. But, it's even worse than that, because the haystack is constantly shifting around. We're dealing with immense computational bloat and batch-specific noise. Biological data is just incredibly noisy by nature. When researchers try to computationally model how genes regulate each other, they often end up with millions of hypothetical connections. The problem really isn't generating the data anymore, it's figuring out which of those millions of connections are actually causing the cancer.
So here's our roadmap for this explainer. First, we'll look at decoding cancer genomic networks. Then, we'll meet the RegNetAgents framework itself, see how it's powered by LangGraph architecture, and check out the MCP integration layer. Finally, we'll look at the proof in the data and the next steps for researchers. All right, Section 1. Decoding cancer genomic networks, and specifically the challenge of cross-referencing datasets. To really grasp the macro problem in these cancer genomics pipelines, we got to look at the data itself. To find robust cancer drivers, researchers just can't rely on one single type of data. They have to cross-reference bulk transcriptomics from TCGA, which basically gives us this broad, tissue-level view of a tumor, with single-cell data from GREmLN, which gives us that ultra-high-resolution cell-by-cell detail. But as you can probably guess, trying to align these two massive, totally disparate networks, it is computationally heavy. Plus, it's super vulnerable to batch-specific artifacts. That's when something looks like a real biological signal, but actually it's just a weird quirk of how the data was collected or processed. Moving right along to Section 2, let's meet the RegNetAgents framework and see how it automates dual network classification. To solve this massive dual network headache, researchers built RegNetAgents. Now, it's absolutely crucial to note that this framework is a downstream analytical validation layer. It assumes you've already got networks built by existing tools, like Arachne. From there, when you feed it a focal gene, it executes this beautifully deterministic pipeline. First up is network retrieval, pulling those pre-computed regulatory edges from both the bulk TCGA and the single-cell GREmLN sources. Second is OncoKB filtering, so it cross-references those candidates against known cancer gene annotations, basically asking, are these guys known bad actors? Third is mode of action, or MOA assignment, to figure out exactly how these genes are interacting in the tumor environment. And finally, consistency scoring. It ranks the outputs based on how much evidence actually overlaps between the two distinct networks. Okay, Section 3. Powered by LangGraph architecture, structuring those analytical steps deterministically. If we zoom into the micro-level software mechanics here, this entire pipeline is orchestrated by a modern AI tool. It's awesome proof that multi-agent frameworks are finally stepping out of those flashy tech demos and into rigorous scientific research. The whole system is built on a LangGraph DAG workflow. DAG stands for Directed Acyclic Graph, which is really just a fancy way of saying the workflow moves forward in a highly structured step-by-step path. No getting caught in infinite loops here. By using this LangGraph DAG, the system perfectly isolates the analytical logic, and this is a real game changer because it completely prevents context window bloat. The raw network data never actually enters the AI's reasoning loop. The AI agents just orchestrate the tools that do the heavy math, rather than trying to, read millions of gene connections in a single prompt. It ensures everything is fully reproducible and fault-tolerant. Next up, Section 4, the MCP integration layer and how it safely connects external agents. So you might be wondering, how do external AI agents safely access this highly structured analytical pipeline without completely breaking it? Well, as the official documentation puts it so perfectly, think of MCP like USB-C port for AI applications. It's essentially a universal adapter. It connects AI models like your Claudes or ChatGPTs directly to external tools and data sources, and it does it seamlessly and securely. Let's dive into this specific implementation. Why did the creators go with an MCP client for RegNetAgents? Well, by routing all those regulatory queries through the MCP client, external agents can fetch these highly accurate cancer driver classifications exactly when they need them. It totally isolates the analytical logic from the AI's reasoning context, which prevents a massive waste of tokens. But honestly, most importantly, it reduces the hallucination risk to basically zero.
The AI isn't just randomly guessing relationships from raw graph data, it's simply querying a deterministic, mathematically sound pipeline through that universal USB-C connection we just talked about. All right, Section 5, Proof in the Data. Let's look at the statistical enrichment of cancer drivers. Zooming back out to the macro impact for diseases like breast and colorectal cancers, we really need to highlight the massive statistical enrichment of OncoKB annotated cancer genes here. The researchers evaluated this across 11 breast cancer and 12 colorectal cancer focal genes. Just look at these Stauffer Z-scores. For the TCGA-derived networks, we're seeing scores of 6.69 and 6.95. And for GREmLN, it's 5.51 and 7.06. Now, if you know statistics, you know those are absolutely massive signals. Literally, every single p-value here is less than 0.0001. That is hard proof that the regulators this AI system is flagging are heavily enriched for actual known cancer drivers. But in biology, you always have to double-check for background noise, right? Like, could the system just be flagging genes that are generally highly active? Well, here's the crucial point. Control sets, specifically housekeeping genes and non-driver genes. They show absolutely zero enrichment. Zero. This definitively proves that the framework is isolating true oncogenic signals rather than just picking up on general background transcriptional noise. And because it compares two completely distinct networks, the system also does this really smart consistency labeling for the candidates. If it gets a label of both, that's your highest confidence signal. A TCGA-only signal often points to bulk averaging artifacts or maybe broad tissue-specific behavior, while a GREmLN-only signal, that can actually highlight rare cell type regulators that bulk sequencing just totally misses. This structured output is an absolute goldmine. It helps bioinformaticians figure out what's a true signal and what's an artifact, way before they spend thousands and thousands of dollars starting wet lab experiments.
Finally, Section 6, Next Steps for Researchers, Implementation and Hypothesis Generation. So how can developers and computational biologists actually start using this architecture today? We've got a straightforward implementation checklist for integrating RegNetAgents as a post-processing step to filter out all that noise in your existing network pipelines. First, pin your LangGraph and MCP client dependencies. Second, make sure you're feeding it pre-computed Arachne networks. Again, this doesn't replace your network inference tools, it just validates them. Third, use the provided Python API to pass your focal genes in and trigger the DAG workflow. And finally, if you're operating in an AI agent environment, route your regulatory queries strictly via the MCP client. That'll protect your token limits and make sure your data retrieval is absolutely pristine. We've really seen today how applying a deterministic AI orchestration to super messy biological data can yield crystal clear insights. As you review the preprint to explore those extended evaluation modules for druggability and clinical relevance, I want you to ask yourself, how are tools like LangGraph and the Model Context Protocol going to accelerate your future biological hypotheses? I think the era of drowning in genomic noise might literally be coming to an end. Keep learning, keep exploring, and I will catch you in the next explainer.